Thank you.
All right, next is Jérémie from Delft University of Technology, and he's speaking about genome-wide association studies done in a privacy-preserving way. All set?
Yeah, I have another one. Yeah, so good morning, everyone. My name is Jérémie Decouchant. I'm an assistant professor in TU Delft. I'm going to talk about the work I did while I was in Luxembourg with my PhD student, Tullio Pascoal. And it was also a joint work with Antoine Boutet and Marcus Wulp. In this work, we look at genome-wide association studies, and our goal is to release statistics on a set of genomes. And we want to do that with a privacy-preserving way. So genome-wide association studies, or I-GWAS, they are studies that look at genetic variations of a set of genomes, and the goal is to identify which genomic variations are responsible for a disease or some kind of attribute. So they can be used for cancer, diabetes, Parkinson's disease, but also for medicine and drug response studies. The way they work is that they're going to study statistical differences between two populations of genomes. So case population where genomes, they actually have the disease, and a control population that does not have it. And they're going to identify the genetic positions that mostly differ between the two populations. So here we have a case population where in some positions in the genome they mostly have an A, and well, in the control population is mostly a T, so maybe that genetic position is actually important for that disease. So these studies, they need a lot of genomes, and the more data you have, the more accurate your results are going to be. So this is a reason for federated environments, but also for dynamic releases and updating those results. Sequencing a genome is expensive, so as soon as you have new genomes, you want to be able to update your results. But updating the results, it's very difficult if you want to maintain privacy. So very quickly, how do we encode populations? So we have what is called a synthetic reference genome. So it is a synthetic genome. It's not a real individual. And in each position, there is a default SNP. So SNP is SNP here. That's the name of a single letter variation in a genome. And for two individuals, you have this kind of well, this is what you have in your DNA. You have two versions of each at each position. And if it differs from the synthetic genome, then we're going to say it's a 1, otherwise it's a 0. And we do that for all positions in the genome. And in the end, we have this kind of encoding here. For each genome, we have either a 0 or a 1. And well, that's the way we encode a population. So in the end, it's just a matrix of zeros and ones. Yeah, so if we look at this I-GWAS workflow, initially you have to collect a lot of genomes, and you sort them in case and controls, and you have your two matrices. Then you want to compute a bunch of statistics on those. They can be aggregate statistics, so for example, just counting the number of ones you have per column, or the number of 1 that you have across two columns. But it can also be test statistics like p-values, r-square, chi-square. And eventually you want to release your results. Possibly you're going to look to release multiple results, and this is the data that researchers and doctors are going to look into. So our goal is to do these kind of studies in a federated way. We have multiple data centers. They can be hospitals, research centers, whatever, and they are going to put in common their genomes. And we want to be able to compute statistics over the whole set of genomes and release them in a privacy-preserving way. So we want our studies to be dynamic because new genomes are always being sequenced. And sometimes we also assume that because of GDPR, some individuals are going to say that they don't want to participate anymore and their genomes should be removed from the study. We also consider that data sets can be interdependent. We believe that it is a sound assumption to assume that it might be possible for one genome to appear in multiple places. Simply because, well, sometimes, communication is not perfect, and also individuals might, get sequenced privately, etc. The adversary we consider can access the results. The I-GWAS results, we assume that they are going to be made public, so we want to make sure that, nothing bad can happen if an adversary has access to that. And we also assume that up to F data centers here can be controlled by the adversary. So the adversary is honest but curious, and it's going to try to launch membership and recovery attacks. So with a membership attack, the adversary has access to a genome and is going to check if he participated in the study. And then if this attack succeeds, then you know that this individual has cancer, for example, which is pretty bad. And recovery attacks means that the adversary is going to try to recover all the genomes, which is equally bad. So if we look at existing works, there are several categories. So first, you can do the computation in a secure, privacy-preserving way. You can use secure multi-party computation, homomorphic encryption. You can use trusted execution environments. But for all of those, only the computation is protected. The release of the data is actually not. So if you want to protect the data that you release, then you can add noise. You can use DP. But then we don't have these other attributes that we want to have. It's not collusion resistant by default. It's not dynamic, or at least, okay, we can talk more about DP if you have questions. The discussion is actually a bit longer than that. We have our previous work here that was also published at PETS in 21. And there, we looked at everything except interdependent studies. We just assume that we have one nice, isolated study, and we release the data. We remove that assumption because, well, to get something more realistic. And this is what we do in this work. So we build on our previous work. We also use trusted execution environments. We could have used homomorphic encryption or secure multi-party computation. It's not the core of the contribution here. So the overview is the following. We have data centers. They collect genomes. They encrypt the data. They send it to an enclave. So we have one trusted central trusted environment that's going to receive encrypted data from the data centers. It's going to decrypt those data in this enclave. It's going to process it and decide, compute the statistics and decide when they can be released. So sensitive data is either hosted on the data centers or in this enclave only. And eventually those results should be released to the public. So a simple observation that we had is that, of course, we cannot update or release the results each time a new genome is created, or each time a genome is removed, because then, you just do the difference, and you have, isolated one genome. So instead of using differential privacy, what we use are, we based our work on a previous paper. And what they did is that they evaluate the theoretical complexity of recovery attacks, and basically it means that you have to use sufficiently enough genomes, depending on the number of positions you're studying, so that it's very hard to launch a recovery attack. And for inference attacks, what we do is that we study, we use a likelihood ratio test, and this is an upper bound on the efficacy of membership attacks. After this test, what happens is that we decide that some SNPs actually, or some positions cannot be released to the public. So they have to stay private, and that's what, so basically they stay in the enclave. And we build on this work, and we extend to dynamic data sets and interdependent privacy. So we have data sets that can intersect. So the first one, if we look at recovery attacks, the only, this condition that we had, this previous work here, is just saying that n is the number of genomes, L is the number of positions in the genome that we want to release. And if we satisfy this equation, then it's very hard to launch a recovery attack. And we extended it before, in the previous work, to tolerate dynamic releases. So we came up with those two very simple to verify equations. They are not optimal, but they are very fast. The other approach that you could have is to do brute force combinations of all possible releases that you have, and make sure that this condition here is always enforced. But that's okay. It does not scale very well. So this previous work here does not tolerate or does not support multiple studies. And now we want to extend to this scenario here, or more complex ones, where you have two studies and they might intersect here. They might look at a common set of positions in the genome, but also a common set of genomes here. So that part here, it appears in both studies. And what we do in the paper, I'm not going to show the result because the formula is a bit complicated, but it's basically theorem number one. And we established a fast equation to make sure that you always have enough genomes that participate, so that you cannot launch recovery attacks easily. This experiment here shows that here you actually need more genomes when you have interdependency. The red one here, so here you have the number of shared SNPs between two studies, and here's the number of genomes that you should use. So the previous formula is the red one here, and when the overlap grows, depending on how you combine the two studies, then you might need actually many more genomes to actually protect it. So if you don't take into account that there might be overlaps or that there are overlaps, then you are going to protect or to release statistics here, and this is actually well, you have this gap here that allows the adversary to maybe recover the statistics. Yeah, and to protect against membership attacks here, the verification unfortunately is a bit complicated. We do have to combine all possible releases, and sometimes we are going to decide that some positions can be released to the public, and sometimes they actually cannot. And then when we have multiple studies that intersect, then the combination is even bigger, and we still have to do all these combinations. So this part is the part where we actually don't have something that scales very well. So we did experiments with simulated and real genomes. We used two data sets, one data set of 2,000 genomes, another one of 20,000 genomes. We used real code, we used IntelliSGX, and we measured various metrics and well, like the release coverage, runtime. Yeah, I won't have time to talk about everything. One important thing here, something interesting is that the number of positions here that we can release is actually quite low. It's 20% here, and it only decreases if you have more positions if you're studying more positions in the genome. We also have a comparison with DP. I won't go over all the numbers, but basically what it says is that DP performs worse with an increasing number of releases, and our solution is, we can compare the two, but we obtain a score or utility of 66 here, and DP here is doing lower. Yeah, unfortunately, I will skip the rest of the discussion here. We also have something that is very fast. That's a red line compared to the brute force that is, an exponential curve. Okay, so I'm reaching the end of my presentation. What we did here is to study interdependent and dynamic releases of GWAS. And, well, we have a better utility and privacy threshold than previous work, but obviously there is still a lot of work to obtain something better. Thank you.
Thank you, Jérémie. Thank you. Questions, and I think I'll hand over the mic, actually, rather than... No questions online? Any questions from the room? I do have a question for you, Jérémie. Actually, I'm just wondering about how would that have been if you've considered the contribution from a fully homomorphic encryption? Just a sort of...
Yeah. So basically, here what we do is that we use a TE to accumulate the data and compute the statistics. If you wanted to use homomorphic encryption, then, well, it would have been more complicated to check for the conditions that we verify. But doing the actual computation is something that can be done. I mean, if you look at previous works, so that's easy. But checking the conditions, that's the part where it would have been more complicated. In particular checking the number of genomes to use, it's easy. This here is not so easy, and I can anticipate, because, well, it's kind of a brute force verification. And with homomorphic encryption, obviously, the cost will be very high. I don't think it cannot be done. I just think performance is going to be not so good.
And while Mina is setting up, I think the other question I had was about the trusted environment itself. Yeah. How tough was it to kind of have that up and running from an implementation point of view?
Yeah, well, for this use case, it was not too bad, actually, because, if you look at the way...
I can't see the slides.
Yeah. Okay, I forgot about that. Yeah, if you look at the data we had, it's only, binary matrices, so it's quite small, and it's actually not too bad. So we don't have to swap, memory in and out, so it was okay.
Okay, thank you. Thanks, Jérémie. Let's thank Jérémie once again, please.